Summaira, come on up. So Alan was really in charge of most of the field breeding activities for a good number of years and shared those responsibilities with lots of others and consul postdocs and graduate students. Meanwhile, Summaira was mostly in the lab and torturing everyone. Faster, more, well, this, but not really torturing very much. But she was really much more in charge of making sure that lab ran properly and whether the marker program progressed well too. And right behind her was Wrong. Wrong, you don't get a chance to speak, but stand up, please. Yay, Wrong. She's truly amazing. If you ever need an exceptional employee for almost anything, go talk to Wrong. You can also make great jokes. So, of course, her name was Wrong Who. And for a while, we had another Chinese woman in the lab named Wrong Who. And so you can make all these lovely little jokes that she never understood or found amusing at all. Can't imagine. Take it away.
Thank you. Thank you so much for Andy for selecting me to talk about my experience in Walker Lab. So I joined University of California, Davis in Viticulture and Enology department to start my PhD in '94. And Andy has been in my entire, when I was a student, he was in my qualifying exam. That was a nerve-wracking experience. But he assured me that we are not there to fail you guys. We are there to pass you. And I took his advice, and it went well. He was in my thesis committee. And then when I finished my PhD, and I went to his office, and I said, well, Andy, I finished my PhD. I need a job. And he said, well, you're hired. And that's it. So rest was a history. And I never looked back and moved away. So in 2002, I joined Andy, his group, and I had opportunity to work with amazing multiple generations of graduate, master's and PhD students, and made acquaintances, made long-lasting friendships, and I hope, and I'm very happy to see many of the familiar faces today in this crowd. So joining the Viticulture and Enology department is like a very historically thing for also that there is a lot of history. Violetti to Dr. Olmo, and then Andy. So I am the third and fourth generation of the grape breeder trained in this department. So the middle picture, I think some previous, I think people have leased. So many of the pictures which I am going to show, Dan shared the pictures with Lise and with Cecilia. So spoilers alert, we, like minds, think the same way. We end up selecting the same pictures out of a whole bunch of pictures. So we are sharing many of the same, showing many of the same pictures. So the middle picture, that's from 2003, when we were trying to figure out the actual, the cross, the 89 series parents, actual parents of 89 series crosses. And so at the time, we visited Dr. Olmo in his assisted living, and only Andy was able to understand what Olmo was actually explaining and describing to him. So we finished, we sorted it out, but that was, I was seeing Dr. Olmo in the hallways of Wixon Hall from '94, but until 2003, and it was amazing opportunity to actually see him. Even in 2003, he has been retired from the department. His apartment was filled with filing cabinets, and he was still working on multiple manuscripts. So amazing legend. And then I had to work with Andy for 20 years, and that has been immense experience. So more recently, just in starting February, I joined USDA ARS breeding program, which also has a rich history. So next year, into 2023, it will be a 100-year celebration of history of grape breeding program at Parlier. And so there also we have big names like Snyder and Loomis, and David Ramming is here. I'm so happy to see you here today. And I have as selected as a grape breeder. So previously, in Andy's lab, I was working as a wine grape breeder, and now I will be doing table grape and raisin grape breeding. So for me, I am in a best possible situation where I'm working, I have to be, and I will be working with developing new cultivars for Pierce disease and powdery mildew resistance. Mostly that's going to be the focus. And I'm hoping to actually achieve and accomplish what I have learned from my predecessors and move the program further forward. So when Karen asked me to give this presentation, I said, okay, what's past, present, and future? And I thought about it, I said, well, in 20 years of my time period, I'm going to see that these are my observations, what I have seen and how Andy has impacted my thought process and how I perceive different things. So what I have observed that Andy is very committed. His commitment to his students, and especially he loved teaching one-on-one series class. He loved interacting with students. Lise, Carl, other students I mentioned, Claire mentioned, all the students who got to do a TA-ship for those classes, they know that Andy is hands-on and he loved enjoy teaching that one-on-one series class. And his commitment with the students, like providing them in proper environment, and also it was his commitment to the industry needs. So as Glenn mentioned earlier, that wherever vineyard you go, Andy already know the people. So that is actually very true, because I have watched him putting a lot of time meeting with the farm advisors, extension specialists, reaching out, giving talks. So that is how he had his fingers on the industry pulse to gauge what are the issues and the problems, and then find the solutions and tune his research program to find the solutions for those issues. So I think that's very important, and that is a lesson for me also, to be involved with the industry as a grape breeder in Central Valley. So in this way, I know that what are the issues and the problems, and then I tailor my research accordingly. So Andy's commitment to his students, as Lise earlier explained in very detail, that it's not just providing them support in academics, it's providing them the nurturing environment. And he opened his house, and we had these legendary Christmas parties, white elephant parties, where we are exchanging the gifts, and you see very young versions of many of us in these pictures. And he was always a child magnet. He was very good with kids, and I think these are mostly Samuel's kids. They are all grown up, now. And Dan's, Anya and Aspen. And then we have some of the historic presents, like Nina holding that white elephant, which got broken down in the same party. And then, and then definitely Kristen's, this picture is, I think everyone selected to show that picture. And then, of course, we have our famous live present, Rooster Jerry Seinfeld. So that was after that party, Andy declared that no one is bringing live animal in the party. That was the end of it, because it was very interesting. It was a big box, and the rooster was crowing. And he said, "What's in there?" And it's like it was Rachel who brought that present, and Rachel said, "Just don't say anything." I said, "Okay." So Cecilia was the one who picked up that box, and she opened it, and she closed it right away. And her face, and her expression on her face, it's a rooster. And then after that, I think that rooster, it has a home in our greenhouse, near greenhouse, but because it was crowing so much, the greenhouse management, they told us that we need to take care of it, and wrong took care of it. I don't want to show you the picture, but wrong took care of it with a big machete. And then, of course, when we were in Vixen Hall, we had very many birthday parties for pretty much everyone. Food was essentially the center of our all activities. Many Dan blowing candles, and of course, so the Walker Lab, at least in my entire time period, and even before me, it seems to be like a family, big family atmosphere. And everyone, every new student who joined they felt that warmth, they felt that comfort, and they joined the group, and they were able to carry out their research projects. And of course, Andy is right. I was the pushing force behind them to keeping them on track. So we were always playing good cop, bad cop. He was a good cop, I was a bad cop. And then, yes, Claire showed some of our collection trips, some of the pictures. So the reason for me to talk about that is that his diligence and persistence, that is the most important thing. Before Claire, like collection trips with Eric, John Rule, Alan Kravanek, we forgot to mention him. He did some collections too, Peter Cousin. So all these, his determination, his focus, his desire to gather the plant material, and especially for the southwestern germplasm, once we learned that from almost initial material, once we learned that it's a very good reservoir for gene material for disease resistance, for nematode, for Pierce disease, for chloride exclusion. So we wanted to make sure that we capture most of that diversity. And in most of those trips, we were driving. I joined Claire and Andy, I think, in one trip. It was Arizona and New Mexico. And we saw lots of wildlife. I had my first exposure with rattlesnake, there was so much other noise from the cicadas. You could not hear him that he was making the sound, and Andy, so I was like 2 or 3 feet away, and I said, get away. It's like, but it's okay, I just want to take a nice picture. And they said, no, get away. So apart from that, there was big giant spiders. And then, of course, we have also the dead horse. I know the Claire, this is a better shot. So on that day, we were driving for almost 3 hours. And we have not seen a single specimen of wild grapevine. And Andy have been to many of these places before, and so he knew that there should be some material. But this was after driving for 3 hours, this was the place where we scouted some grapevines. So before I get into the story, when I joined this collection trip, I was not sure what they are looking outside the window, what they are looking at. And then Andy, he said that you have to train your eyes to look for grapes. And lo and behold, once you know how to focus and how to look, then you see wild grapes everywhere. Wherever you have some water, you are bound to see some wild grapes there. So for this dead horse thing, that was our first sample. So both Claire and I, we were the one to get out of the car and gather the sample. So, and we collected, I think, three or four samples, because it was not just one plant, it was a grove of plants, and they could be the same or they could be the different, so we had to collect the specimens from all. So they end up quite diverse. So in Walker Lab, we got the opportunity to work with a wide range of pests and pathogens. I was mostly focused with powdery mildew and Pierce disease resistance breeding, but I was also involved with the rootstock. So it was this big project, running it and making sure that we have enough funds. So he shielded us from all the administrative part so we could focus on the science. And thank you so much. Now I'm in a practical job, and I can see that how much you have shielded us, and because there are some moments of frustrations when you want to go back to the old time when you can just focus on research and you don't need to worry about the writing proposals or writing reports or getting doing trainings. So thank you so much, Andy, for that. So it depends. I think Carla mentioned that phrase. So wherever you go, and I think Jean also said a little bit, whenever you go to Andy, you are not going to get a straight, clear answer, and that's actually true. Philosophically, if you look at that, each problem and issue has multiple sides. And it's your answer, it depends on which side and how you're looking at the issue. Most of the time when I was going to Andy, I was asking him regarding that, okay, if this is a hybrid of Vitis candican, like B4317, it's a complex hybrid, and every time the species would change. And it was just like, his answer would start always, it depends. It depends what's your perspective, it depends how you want to look at the issue. And what I learned from him also, that smile with a positive attitude, it can take you far further in your career compared to if you are not in a good mood all the time. And I have seen Andy always happy, and most of the time when he come to the lab and you see him, or when you go to his office, and this has been echoed by many of his students, that he is so available to his students, and with a positive attitude, to get and doing the things. I also learned from him, simplicity is the best way for any approach. Nature works in a simple way too, and working hard. So what I saw Andy, and I'm actually going to be following in his footprints for that, working hard. He was in the office 7 a.m. and he's leaving at 5 or 6 p.m. So by working hard, I think you can overcome all the obstacles. And actually, that was the way he was able to handle so many graduate students and work on so many projects and make sure that everyone is provided for in the lab and we have the right environment and right infrastructure to carry out all the research. And then he was very observant and attuned to the industry needs, and that has been apparent. Like, he was involved for traveling a lot, meeting with the growers, nationally and internationally. And I think that's a very important thing for the new grape breeder as well in the department, that he needs to follow some of these traits if he wants to be successful. I wish him all the best for that. And you can work hard, and by the end of the day, if everything is not complete, tomorrow is another day. So that was also the philosophy of Andy, and I think that it's a simple thing, and it helps a lot to make a bad day into a good day, because tomorrow is another day. And thank you so much, Andy, for providing me the opportunity to work with you and learn a lot, and work with all the graduate students. And I hope that what I learned from you, I'm going to take on to my job at USDA and be successful as you have been. Thank you so much.
Okay, that was mostly true also. Not all of it, but it's getting closer.